Okay, I will assume that you can see it. So thanks a lot for giving me this opportunity to talk to you today about iSEE, which is an R package built on the Shiny framework that lets you construct flexible interactive applications where you can basically explore any type of rectangular data, including biological data, and also share these insights with your colleagues. So before showing you what iSEE can actually do, just a word about the input data. So the data that you provide to iSEE needs to be in the form of a so-called summarized experiment object. So this is a general class for storing rectangular data from the bioconductor project. And you can also have annotations for both the rows, so the features, and the columns, so the samples. So if you have this type of rectangular data, it's actually easy to create one of these summarized experiment objects. And I should also say that any type of class that extends the summarized experiment will work as well. So for example, the single cell experiment, where you can also include predefined reduced dimension representations of your data into the object. So let's see what iSEE can do. And first of all, it's really easy to get started. If you have a summarized experiment object, these 2 lines of code is really all you need. You need to load the package, and then you need to call the iSEE function with your summarized experiment object. So here we show an overview of an example applications. And in this case, we're actually looking at single-cell RNA-seq data. But as I said, any type of rectangular data really would work just fine. So iSEE is based on the concept of these panels that you can see here in different colors. And the idea is really that each panel provides a different view of your data or shows a different aspect of the data. So the iSEE package provides 8 of these panel types. There is an add-on package called iSEE, which provides additional panels. And you can easily create your own panels if you want to perform specific analysis or make specific visualizations. So the basic iSEE panels don't actually perform the analysis internally, let's say. They don't run the PCA or run the clustering, but rather visualize the data or the information that's stored in the input object. It's totally possible to build panels that perform analysis, but having the basic ones just visualize the data that's in the object have some advantages. So for example, we're not locking anyone into a specific workflow. You can analyze your data in any way you want using the same tools as the same pipelines that you're usually using. Where it also means that you don't have to wait for lengthy calculations when you do the interactive analysis, which also makes the package a bit more scalable. And in fact, you can make it even more scalable if you have like many thousands, tens or hundreds of thousands of cells, for example, by only displaying a subset of the points, or even working with disk-backed data like HDF5 files. So part of the flexibility of the iSEE interface comes from the fact that you're totally free to define which panel types you want to include, how many of them, and in which order they are displayed. And this can be done either programmatically when you launch the app, or interactively as shown here. So you can remove some panels, you can reorder the panels, you can resize the panels, and you can add additional panels. Everything to make your visualization, your application look optimal for your type of data. And in fact, looking around at some different apps that are using iSEE in the wild, so to say, you can see this diversity. You can see that these authors have used different combinations of panels to convey their message. So once you have set up your app, you want to start exploring your data. And one way that iSEE provides for you to do this is to color the points according to the provided annotations or observed values, for example, gene expression levels. So here we have our cells. We color them by the cluster annotation we have in the object, or the cell type label, or we can color them according to the expression level of some marker genes. Then another way of interacting with your data is by making the panels talk to each other. So for example, in this case, we're selecting some cells here in the first panel, transmitting the selection to the second panel, which automatically finds the genes that are most highly upregulated in those cells. And then we select the top 10 of these genes and make a heat map out of those. So really just an example of what you can do. You can imagine many other things, I'm sure, like highlighting all the genes from a specific pathway in your volcano plot, or finding where in your t-SNE plot are those cells that co-express multiple marker genes, and so on and so forth. Another important feature of iSEE is the combined focus on interactivity and reproducibility. So inside the app, you always have access to the exact code that you would need to reproduce the plots that you see. So if you take all this code, you copy it and you paste it into your R session, create all the plots that you currently see in your interface. You can also export the code to create the panels that you currently see. So these you can use to initialize the application to start in exactly the current configuration, which can be useful if you have found this optimal configuration you want to use, or if you want to share the information with someone else to want to make sure that they see the same things. And finally, in terms of communicating results to other people, iSEE offers the possibility to include interactive tours that lets you basically walk a user through the different aspects of the interface to tell a story of the data or to kind of help them orient themselves. And importantly, this does not prevent the user from at any time just exiting this tour and exploring things as they want to on their own. So how is this useful? Well, I think there are many answers to that. But for example, it can enable a larger number of people to actually explore their data in depth, to more easily generate hypotheses and detect any issues with the data. It's also a nice way to collaborate and to let other people explore the data from their perspective, which also kind of increases the reach and the impact of the data that you're generating in your organization. And to further increase accessibility, you can, for example, deploy iSEE on a Shiny server and easily set up a custom landing page where users can just browse, select a data set from a drop-down list, for example, and go ahead and directly explore it. So with that, I'd like to round off and acknowledge my fellow co-developers, Kevin, Federico, and Aaron. I've added some links to additional information here, and I would like to thank everyone for your attention.